As I introduced myself, my name is Ekhtiari, University of Minnesota. I'm a psychiatrist and also being trained to be neuroscientist. I attended OHBM first time 2010 when I was an MD PhD student, and I'm still attending this really kind of nice community of scientists. And I'm very happy that we have this workshop discussing about how to use neuroimaging in a clinically meaningful way for potentially treating people with different brain and mental health disorders. So I do not have any kind of conflict of interest. Before starting my talk, I would like to thank my colleagues and collaborators, Laureate Institute for Brain Research, University of Minnesota, and my lab members that are mainly kind of working together to see how we can use brain stimulation to develop new treatments for psychotic disorder. And I'm recruiting postdocs, so if you are interested, we can definitely discuss after the talk. And just an overview of what I'm going to discuss about. So the first talk would be just giving you an overview about the levels of optimization that we can potentially do using different sort of neuroimaging methods that we have, and discussing about 4 levels of optimization: on target optimization, dose optimization, timing optimization, also optimization of the context. And we'll further discuss about that. And then in my presentation, I will also discuss about the new GUI that we put together that has kind of specific pipelines for individualized and personalized fMRI-informed TMS. And you will have access to that GUI as well, that one of my postdocs, Ghazale, is already kind of working on, and you can kind of have resources to be able to use. But today we are here to discuss about the combination of neuromodulation technologies and brain stimulation. Those who are in the room, how many of you have touched any of these brain stimulation technologies yourself? Okay, almost everybody. Okay. How many of you are doing actual patient care with these technologies? Okay, half of the people in the room. That is interesting. That's a kind of an important part of what we are doing in kind of human brain mapping society. In many years, it was not very much. I mean, clinicians have been involved, but it's just the number of clinicians who are getting interested to know brain imaging is just increasing because of the potentials of using that with neuromodulation technology. So there are definitely interesting potentials on that side. And you know the field is just exploding in terms of the number of publications. You know these technologies, the number of studies that people are doing is just increasing. And when we discuss about the approvals that we have, the regulatory approvals that we have in the US for FDA, how many FDA-approved applications we have using TMS in the clinical practice? How many? 2? Okay. 1? 2? How many FDA approvals or clearances that we have for using TMS? For 3? Okay. Okay, so the first one, MDD, October 2008, and then OCD 2018, smoking cessation, August 2020, anxious depression 2021, the new accelerated protocol, we call it SAND protocol, that has individualized fMRI 2022. And I mean, Desmond is going to discuss about different things that he's doing, and Mario and others, and we'll discuss about also new interesting things that are happening in the field in terms of using individualized fMRI as a kind of part of the treatment protocols. And then you know that there is an age extension starting from age 15 to 86 right now that is being approved by FDA. So these are the approvals that we already have in the field. And potentially there are going to be new approvals that potentially are going to be about different substance use disorder, PTSD, bipolar disorder. There are other areas that people are hopeful to be able to get FDA approval in the future. But when you work with, I mean, people and you do these sort of interventions, you see that there are people who are responders, okay? So this is a sample study of seeing people who are responders. And if I activate this guy, okay, so how many of the people in the room, they know what is ITBS 600? Okay, so there are, so ITBS is a protocol that we use in TMS, a specific pattern pulse that we use, and potentially it is supposed to increase the level of excitability that we have in the cortex, starting with motor cortex. This is one of the kind of the first study. It has been published in 2015, discussing about when you increase the dose from 600 pulses to 1200 to 1800 pulses, the level of excitability that you have in the motor cortex would go up, potentially not very much from 600 to 1200, but potentially with kind of higher doses, you have higher response, okay? But you always have a group of people who are non-responders. And the question is, why you have people who are not responding? So, and here we are targeting motor cortex, and with doing that in motor cortex, there are people who are not responders. And the question is why, and how we can potentially address that. So that could be sort of a starting point for a discussion. I'm gonna spend a few minutes on that and thinking together why there are people who are responders and there are people who are not responding when we do that stimulatory protocol over motor cortex. Anybody who is interested to start to share their thoughts? Just one reason.
Just from my understanding, if you talk, Suzanne?
Can I have the mic?
Oh yeah. Is it just like generally, or you mean in specific, like depression or so?
This is specific. I mean, whatever comes in your mind. One reason that people might not be responding.
I would say like brain circuitry is just like generally very different in among people.
So you think that we are not in the right target? Okay. What else?
Stimulation intensity is not enough for some participant.
It is not, the dose is not. So we are not in the right target, the dose not enough. What else?
Behavioral state.
Could you explain further?
Perhaps some participants are anxious, stressed, trying to inhibit any kind of motor response. Maybe they're asleep. I doubt it, but that's what I mean by behavioral state.
Okay. So the context or the state would be a factor for being responder or non-responder. What else? What are the other factors?
I'd say in the bucket of measurement error, they often, in MEP studies, undersample the number of trials you need to get a stable response, and they don't carefully quality control their data to get rid of artifacts, spurious responses.
So measurement. So we have a challenge in how to actually measure the response, and there could be error in measurement. What else? What are the other reasons?
Perhaps lack of plasticity.
Will you explain further?
So if there's some kind of underlying deficit in the sort of plastic nature of their circuitry, they're not going to be, I mean, so these are non-responders to treatment, and you're not seeing a response in their sort of motor evoked potential, so it might be just due to this kind of like the substrate, right?
So basically, the neural response, you are expecting a level of plasticity, right? Then the neural response is going to change over the time with that intervention. And there are some specific molecular mechanisms that underlies that specific neuroplastic changes, and those mechanisms might not be, for many reasons, working in these kind of specific population. And you know, there are even studies that they try to add some medications to open up those kind of neuroplastic gates to be able to have the neuroplastic responses. What could be the other reasons for that? Any other suggestions? Any other thoughts? Okay, so it's always good to start with some questions, right? So now let's just kind of further discuss that and see what are the other potential mechanisms that might be important. And that could be a good question that you can have with yourself through the entire course, and what other speakers are going to discuss about why there are people who are responders and non-responders. And is there any chance that we can make a non-responder to a responder? And is there any chance that we can optimize the intervention in a quality that the intervention would make, we will have kind of responders to that specific intervention? So my talk is mainly around addiction as a clinical population I'm interested to. And as I mentioned, smoking cessation was one of the FDA approvals in August 2020. That was basically based on a deep TMS coil, H4 coil, that is mainly targeting frontopolar cortex. And I will kind of give you examples from my own field of kind of work, addiction. I think that those things could be helpful for other areas that people are working in. And other speakers are going to discuss about other disorders as well. So an overview about what is actually happening in the field of addiction with electrical stimulation and magnetic stimulation, TES and TMS. This is the timeline of the progress that we have in the field. Any estimate in terms of when was the first TMS study in the field of addiction? Year of? 2010, okay. You are cheating based on the graph, okay. So the first studies on nicotine, 2003, on cocaine, 2007. The first TMS study on cannabis, 2018. So the field is still growing. When you talk about direct current or electrical stimulation, the first study on opioids goes back to 2016. So the field is still in kind of infancy or early adolescence. And when you go to the details of other things that are progressing in the field, and one specific thing that the first sort of fMRI informed individualized TMS study in the field of addiction happened in 2023. So individualization using MRI, doing optimized intervention is a pretty new thing in the field. So we are discussing about all the kind of new advances that are happening in the field. And if you look into all the trials that people did for addiction, electrical stimulation and magnetic stimulation, it's over 242 by the end of 2024. So the number is increasing, as you can see here. And of course, nicotine, alcohol, amphetamines, and the number is increasing over time. So this is the current status of the field. So people are working with different trials. So over 240 published trials in the field. And if you put all of them together with the main clinical outcome measure that we have in the field of addiction, the gold standard is consumption or relapse or use of substances, right? And one of the proxy measures of that is craving. So craving and consumptions are two major clinical outcome measures that we have. If you put them together, there is a positive response, overall positive response of electrical stimulation and magnetic stimulation on craving and consumption measures, okay? But the challenging part is, if you look into the details of those studies, what you see here is that those studies targeted multiple different areas in the brain. So they targeted very different circuits and very different kind of areas in the brain. And even when you go to the details, for example, if you go and look into the studies that they, for example, they targeted dorsolateral prefrontal cortex. So these are studies that they target dorsolateral prefrontal cortex. If you go and look to the details of what they did, you can see that they defined DLPFC with very different ways, using EEG, 5 centimeter rule, 6 centimeter rule, MR guided, beam, F3. So there are different ways of even defining DLPFC in the field. So people are doing different targeting. And that is the situation in many other areas in the psychiatry. So people started initially with the idea of basically doing skull-based targeting, using the skull-based measures, or finding the hottest spot on the motor cortex and then coming 5 centimeters ahead of that and defining DLPFC as 5 centimeters ahead of what we have in motor cortex, right? And this is not really accurate, not very precise of way of doing that. And then basically people try to use anatomical targeting to increase the precision while we might not be exactly in the place that we are interested to be. And then the idea of potentially using brain mapping, the tools that we have, using MRI, using the activity-based measures or connectivity measures as what we call kind of individualization or kind of optimizing that with higher accuracy and higher precision. And then now there are discussions in terms of how we can do that in that individual level to do the optimized stimulation. So this is the pipeline that is happening in many areas of psychiatry right now. Depression is the main area that our speakers are going to discuss about, but it also happens in addiction as well. But when you are seeing this, you have basically two levels of target optimization. One is what we call group level target optimization, and the second is called individualized level target optimization. So just starting to learn the differences, we can do group level target optimization, and then everybody would receive this kind of the same target, or we can do individual level target optimization. So these are the two levels of optimization that we will further discuss about. When you talk about depression targeting biomarkers, the field is growing, and we'll further discuss about that today as well. Even there are companies that they started to use those measurements of targeting biomarkers and patent them as a way to be able to make a sort of business model for targeting specific circuits in the brain for depression. And this is an area that is kind of evolving over time. And there is already, as I told you, that the SAND protocol has a specific FDA approval or FDA clearance for using that sort of personalized targeting in the field of depression. But in the field of addiction and many other areas of psychiatry, there are still discussions about what is the right target, how to find the best targeting biomarker. And this is the model that I like more than others in terms of there are multiple networks that are working together when you are discussing about addiction, and potentially the balance between these networks, executive control network, default mode network, and saliency networks, important, what we call as craving. And in the craving state, we probably have higher activation in some areas related to default mode network and also some subcortical areas. And when we are in a control state, we have further activation in executive control network and top-down regulation over the subcortical areas that are involved in developing the urge to use substances, okay? So this is a sort of the model that we are interested to consider when we are discussing about addiction. So potentially there is not one single target. So there are multiple different targets that you can consider within this specific large multi-networks that are interacting with each other to develop the addictive behaviors. Even in the new systematic review and meta-analysis that we did, and we evaluated all the networks and all the protocols that people try to use in the field of addiction, there are 3 protocols that you can see them in the green lines, that they have the highest consistency in terms of the effect size and the the significance of the intervention. One is high frequency RTMS on left DLPFC. And then the other green line is the high frequency of targeting the frontopolar cortex with H4 coil. And then the other one is using electrical stimulation, putting the electrodes on both sides and targeting frontopolar cortex. And these 2 or 3 different protocols, they are targeting frontopolar cortex, as you can see here, or they are targeting DLPFC. So these are the 2 major targets that people try to consider, frontopolar cortex or DLPFC. And there are basically components of the network that I was discussing about in terms of, there is a little bit of delay between, okay. So this is what we call a dual target model in terms of there is a frontopolar target and there is a DLPFC target. And it seems that both of them are eventually interacting with subcortical areas, amygdala and nucleus accumbens. And this is the model that we have in terms of the level of interaction between frontopolar cortex. So DLPFC is sort of suppressing subcortical areas, and frontopolar is engaging those networks. And this is the model that we have that seems supporting the level of evidence that we already have in the field. So this is the way that people are trying to find the right target and optimize the targeting protocols in the field of addiction. To be able to actually do that in a trial, as an example, what you want to do the individualized targeting. This is a kind of a pipeline that we developed to be able to target frontopolar cortex in an individualized way, to be able to find the best way of interacting between frontopolar cortex and amygdala. And this is a population of 60 methamphetamine users that we try to find the best area on the frontopolar cortex to have a window to their amygdala based on different levels of evidence that we had in the past. And as you can see here, this is the space that we have in the x-axis, y-axis, and z-axis. And these are in millimeters. And these are individual, each of those dots are individual participants in the database. And you can see they are sort of widely distributed across different areas within the frontopolar cortex if you want to find the best area to target in the frontopolar cortex. And we basically showed that these sort of individualized targets and their connections to amygdala are related to the level of craving that people experience. So this is the way that we do optimize the location of the stimulation. So you can do optimized targeting based on the location. Many other studies that they are in the field that they targeted frontopolar cortex, they went only for FP2 or FP1. And FP2 is more frequent target in the field. But as you can see here, this is the one-size-fits-all target for everybody if you go for FP2. But if you do an individualization, that is going to be distributed around. And you can do the individualized targeting if you are interested in that specific area. But beyond the location, when you are doing TMS, there is another level of optimization that is going to be important. And this is the coil orientation. So changing the coil orientation is going to change the amount of kind of electrical impact that you are making on the target that you have. And you can even optimize the orientation of coil in a way to be able to engage that target further. And to be able to optimize that, we usually kind of use the robotic arm recently, just making sure that we will be able to find the right target and also the right orientation of the coil. And meanwhile, there is always a challenge in terms of how to optimize the dose, because people, based on this, the size of their skull, the size of their frontal sinuses, they would receive different levels of electricity in their brain. So if you want to make sure that everybody would receive the same dose of electricity in the trial, you need to optimize the dose of the stimulation. And honestly, we'll discuss further in the discussion that we have. I think that dose optimization might even be more important than the target optimization. And sometimes people completely forget that piece. So dose would be a very important factor that we need to make sure that we are going to optimize. So the first level, if you want to name that level of optimization, how do you name that level of optimization? And also the second one. Is a group level optimization or individual level optimization? It's an individual level optimization. And also the first one is the individual level target optimization, and the third one is individual level dose optimization, right? So these are two levels of optimization. You can optimize in the individual level the target and also the dose. So that specific pipeline is published in Addiction Biology and is already available. And then we put all the analytic pipelines that we have and the processes in a GitHub. And the GitHub page is already available. And I shared all my slides in the QR code that I had in the first slide. And also I will share the link to my slides to the WhatsApp group that we have. So we'll have access to the slides. If you want to have access to the slides, you will have the access to the slides in the the WhatsApp group. So the idea of individualization could go for frontopolar cortex, for DLPFC, even within sort of dual target, we can individually target those dual targets. And also now we started to go further and look into other targets in the parietal cortex to see how we can find the right target in an individual level in the parietal. So there are definitely other opportunities for that level of individualization. Just one level further to kind of wrap up the discussion that we have, just to give you an overview, and we are going to further discuss these things in different talks. So one of the other ways of individualization is what we call closed loop stimulation. In terms of being able to record and do the stimulation and see how our stimulation is changing the recording that we have, and then optimizing that over time in an individual level. So that is another level of individualization that can individualize multiple different factors inside the scanner. To be able to do that, one of the areas that we are interested is doing electrical stimulation inside the scanner. And it's an interesting area because it seems that the electrical stimulation could be happening inside the scanner if you can manage that well inside the scanner. I mean, Desmond and others and Mario are going to discuss about doing that with TMS as well. But with TMS, it's more challenging. There are lots of other technical details that you need to consider to be able to do that. But it seems the electrical stimulation seems more doable inside the scanner. So we invited all the labs around the world that are doing electrical stimulation inside the scanner. And we asked them to share their experiences and ask them in terms of how we can collaborate together to develop quality standards. Because when we have been reviewing all the literature in the field, we realized that many labs didn't report the details of how they actually did the stimulation. So we asked everybody to say, okay, if you want to do a really good quality electrical stimulation inside the scanner, what are the factors that you need to report, okay? And we published a checklist in Nature Protocols about concurrent electrical stimulation fMRI, and we are asking everybody to follow that. If you are interested to do electrical stimulation inside the scanner, please kind of use that sort of checklist, making sure that you are considering those factors and also you are reporting those factors in your studies. And this is the kind of the area that I'm interested to do inside the scanner, kind of a nice group of people that we work together. And we even do that while we are exposing people to drug-related cues and see how we can modulate the level of craving that people experience inside the scanner. And the idea of how we can do the optimization with different levels of optimization. We can optimize the electrical stimulation, the frequency of stimulation, or optimizing the phase of the stimulation. So these are the factors that Ines is, I can imagine, will talk about many of those factors. And the idea of doing electrical stimulation inside the scanner while closing the loop in terms of defining a specific target. For example, we are interested to measure the level of connection between frontoparietal cortex and how the engagement of frontoparietal cortex can be modulated with electrical stimulation, different modulation in the electrical stimulation, and how the optimization could be happening. That is the way that we are interested to do. We have been able to make that happen at Lloyd Institute for Brain Research. There are some technical details in terms of how to actually do that inside the scanner, and we put all those details in a technical report. If you are interested, you can look into that technical report, and I'm definitely happy to help others that are interested to follow that. So just wrapping up the discussion and the topics that we have been discussing about today in my presentation, we discussed about different levels of optimization so far. We discussed about target optimization and how to optimize the target using fMRI, using structural-based, even imaging, lesion-based measures, all those measures that could be used for target optimization. And we discussed about dose optimization. And as I told you, dose is definitely very important. One of the papers that I really like that is published in the field of depression, and they explored a large database of over 7,000 patients with MDD, okay? And then they showed that there is a sort of rapid improvement in response to TMS for depression in the first 10 sessions. And then there is a sort of a steady growth in terms of the number of people who are responding, the level of response that they have. And they reported that less than 30 sessions of TMS, you will not get to an optimized level of response. So without doing TMS for 30 sessions, you will not get to a kind of optimal level of response. And that is an important issue in terms of sometimes even there are patients who are late bloomers, and they do not respond in the first 20 sessions, and then you see that they start to respond in the next steps, which is quite an interesting pattern of response in terms of why there are people who do not have any response in the first 10, 20 sessions, and then they start to have the response. And there are interesting discussions about that in terms of how the dose is going to be important. And not only the dose, also the timing of the way that you are providing the dose. So the other area in optimization is what we call, it's called the context. And as one of the participants mentioned, that the context is always an important factor. And the reality is when we do the stimulation, depending on the connection of the node that we are targeting in a network level in the brain, the connection that the node has would define the level of response that you have or the type of response. So if you are in a state A and your node is connected to specific areas compared to a state B that your node is connected to other areas, your response would be different. So that is an important issue about the state. And even the FDA approvals that we have for addiction and also for OCD, they have a component other than TMS. Does anybody know what is that component? There's a specific behavioral component that the protocol is approved to do TMS with that behavioral component to be able to put people in a state to be more responsive to TMS. What is that? Basically, it's a Q exposure protocol. So for OCD, we expose people to things that would be related to their OCD behavior, and also for addiction, it's just Q exposure related to substances. So the provocation or the Q provocation is a part of the protocol that we have there that will put people in a state that they will be responsive. But when we talk about the context of the state even could be a chemical state. And you know, there are studies even they show that things like kind of NMDA kind of agonists, I mean, things like D-cycloserine, things like that, they could change the level of response that people have to brain stimulation. And then the last level, discussing about the timing of the stimulation and the factor of time. and how to optimize the timing. And colleagues will discuss about that in terms of how to optimize the stimulation in a time domain. And the time domain could be in milliseconds or in weeks and years of the stimulation. One specific example, we are, as I mentioned to you, we are interested to frontopolar ventromedial prefrontal cortex, DLPFC, ventral striatum network. And we showed that kind of this specific network is changing during the time of your substance use. If people are using substances over time, the connection these networks is changing in terms of, for example, in time, the connection between ventral striatum and VMPFC is going down, while the connection between VMPFC and DLPFC is going up. And even if you divide your patients into two groups of, for example, those who are, this is methamphetamine user patients, those who have methamphetamine use between 0 to 7 years, and those who have the methamphetamine use between 7 to 14 years, and between 14 to 22 years. So if you divide them in three groups, the type of connections that they have in their brain in response to drug-related cues and craving is different. And that could also inform potentially the target that you want to consider. And that is another important factor in terms of, okay, in this specific scenario, potentially, this specific connection is going to be more important for me compared to other connections. Okay. The last point that I really like to mention as a sort of another thought-provoking issue is that at the end of the day, whatever we are doing with neuromodulation, we need to make sure that people in the community would have a good understanding about what we are doing. And in the society, people understand about the importance and the way of the stimulation, how the stimulation works. And honestly, when I do TMS trials, I usually receive the intervention that we did design myself in a lower dose to just, first of all, experience what we are doing, and also make, the safety and whatever you are going to do for your patients and your participants, you are supposed to be happy to do it for yourself. So it is important to do that. But meanwhile, when I work and discuss with people in the community, especially that the marginalized communities and unprivileged communities that we have in the field of addiction, what people think about kind of brain stimulation is the picture that they have from One Flew Over the Cuckoo's Nest. So that is the sort of the kind of community understanding about brain stimulation. And it's always important to engage communities in all those advanced robotic-based, individualized, highly kind of sophisticated brain imaging things that we are doing for neuromodulation, to make sure that they know what we are actually doing and how this is working. So we developed a specific network in Minnesota. We call it Minnesota Innovation in Recovery Network. And whenever we design a new trial with advanced brain stimulation and advanced brain imaging technologies, we have people from the community as a part of the board involved in that process, showing what we are doing, even discussing about how we are optimizing those things, and making sure that they have a say in that process, and also they share their ideas. And to be able to do that, we are also developing multiple sort of cartoon-based materials about brain and how brain stimulation is happening. So we are extending those materials for people in the community. And now people start to translate them to different languages. We already have them in 22 different languages, some of the sort of neuroscience-informed educational materials. And now we are extending them to a specific different areas of brain stimulation, including TMS, electrical stimulation, and we hope that they are going to be an important part of what we are doing. Okay, so take-home notes. Going back to the issue that I raised in terms of the responders versus non-responder, we can definitely further discuss that during the workshop today. So today we discuss about circuit-based neurotherapeutics, and we discuss about group-level target optimization, individual-level target optimization, dose optimization, timing optimization, context. There are definitely interesting cross-species dialogue between kind of animal models and what we do in human models, neuroscience-informed psychoeducation, and also global collaboration. People in the room, you are from very different backgrounds. I mean, people, when they have been introducing themselves, I think we have people at least from 30 different countries in the room right now, which is fantastic to see. And the reality is, even if you look into brain stimulation studies that are happening, especially, for example, in the field of addiction, it is not only in the US. It's just, in the US, we are producing like 20% of research in the field. So 80% of research is happening in other parts of the world. And we realize that we need to bring people from other parts of the world to collaborate together. So we develop a network that we call International Network of Electrical and Magnetic Stimulation in the field of addiction. We have a specific consensus statements that we bring people together. We already have 54 labs around the world that are a part of that network, and different sorts of other publications that we have. We have a YouTube channel that we post the videos and the kind of discussions that we have in that YouTube channel. They're already available. We have, every 2 years, we have a specific panel that we get together, discuss about the future of these sort of activities around the world, and the videos are available on YouTube channel. And there are some ongoing living systematic reviews of the field and other networks and our potentials for collaboration in the field. So this is what I really hope about what we are doing in OHBM, to bring people together and collaborate. And there are specific other international global things that we are doing in this field, and we are hopeful to be able to encourage you to join us in those activities. Thank you very much for your attention, and I will move. Thank you.